Hello, welcome to embryology classes. So today is the first session on embryology. So in this session we are going to see about the cell cycle and cell division. So first regarding the cell cycle is an ordered sequence of events culminating in the cell growth and cell division to produce two daughter cells, and it generally lasts minimum of 12 hours, but in most adult tissues it can be considerably longer, and it is divided into four distinct phases which are known as like gap 1 phase, G1 phase, growth, where we can see the preparation of DNA synthesis. Then S phase is the DNA synthesis phase, where DNA undergoes replication to form a double amount of DNA. Then G2 phase or gap 2 phase, where it is a preparatory phase to the mitosis, preparation for mitosis and growth. And finally the M phase. M is the mitosis phase, which where it is having combination of four phases again. So mitosis contains several phases. So the combination of G1S and G2 phases is altogether known as interphase. And M phase is the mitotic phase, which is further divided into four phases: prophase, metaphase, anaphase, and telophase. So the DNA synthesis, that is the replication of genome, occurs during S phase. So DNA synthesis occurs during S phase, at the end of which the DNA content of the cell is get doubled. So at the end of S phase, we see the DNA becomes double. The genetic material becomes double. The times taken for S and G2 and M are similar for most of the cells, and occupy around like DNS phase 6 to 8 hours, and G2 phase 2 to 4 hours, and mitotic phase 1 to 2 hours respectively. And in contrast, the duration of G1 phase considerably varies. So it is variable, and sometimes ranging from less than 2 hours is in rapidly dividing cells, and more than 100 hours within the same tissue. In ovaries, for example, primary oocytes becomes diplotin for 5th month of intrauterine life, and each of it remains in this stage until the period of ovulation, that is till the period of adolescence in women, and it continues up to the age of 50 years. So we can see the gap 1 phase varies a lot. So in highly dividing cells, this gap 1 phase may be very less, and in a slow dividing cells, the gap 1 phase may be very long, and in the cells which are dormant, just now I explained, like in primary oocytes, the cell cycle may be arrested at a particular phase and kept in a particular stage. So that is about the cell cycle. So next we shall discuss about this mitosis, the type of cell division which is very common in somatic cells. So this type of cell division occurs in the somatic cells, and the mitotic cell division is the process whereby one cell divides into two daughter cells that are genetically identical. So the one cell, it divides into two daughter cells which are genetically identical to the parent cell. So each daughter cell receives the complete complement of 46 chromosomes. So the parent is having 46, the daughter cells, each daughter cell also will have 46 chromosomes. So the period between the two mitotic divisions is called as interphase. So just now we discussed about the interphase as G1 phase, S phase, and G2 phase. And if cell doesn't divide, it enters into GO phase. And so let's see the subdivisions of mitotic division. So the first phase is the prophase. In this stage, the nucleolus disappears, and the chromosomes becomes coiled, the coiled chromosomes, and they start getting condensed, shortened, and get thickened. And each chromosome is now consists of two parallel subunits. You can see the two parallel subunits. So if you see a nucleus in the interface, these chromosomes are highly coiled. It appears to be like a noodles in a cup. Whereas in the prophase, they align themselves and they get shortened and condensed and they form like a parallel strands. So each of these parallel subunits, they are called as chromatids, and which remain joined to each other at a common region called as centromere. So this white part is the centromere, and blue and red we can see the chromatids. But the chromatids cannot be recognized. You cannot recognize these chromatids at this phase. Next is the pro-metaphase. Pro-metaphase, in this stage, the chromatids become distinguishable. You can nicely separate, you can see distinguished chromatids. So that's how you can designate this stage as pro-metaphase. Next is the metaphase. In metaphase stage, the nucleus becomes like breakup. The nuclear membrane will break up. Here we can see the nuclear membrane. Here there is no nuclear membrane. It breaks, and double structured chromosomes they line up in the equatorial plane. You can see they are lining up in the equatorial plane of the spindle and get attached to the microtubules of the spindle, extending them towards the centrioles. So they are extending towards the centrioles, which are present on each opposite pole of the cell, on each one pole of the cell. And the next phase is the anaphase. In anaphase, the centromere of each chromosome splits into two chromatids and are separated from each other, and they are now called as daughter chromosomes. The spindle fibers attached to the centromere, we can see the spindle fibers attached to the centromere towards the poles. So due to the pull of these spindles, the shape of the arms trailing as they move forward the poles. So you can see the arms are opposite. So the centromeres are towards the poles. So they'll start moving in anaphase. Next is the telophase. In this phase, the separated chromatids are now, so you can see this chromatids are separated, and the spindles are completely disappeared. The centrioles are there here without spindles. So the spindles fibers are disappeared, and nuclear membrane appears rounded. Again they surround around the chromosomes. We see the nuclear membrane, and each polar group, the daughter chromosomes are seen, and the chromosomes uncoil and become less compact, and the nucleolus reappear again inside each nucleus, and there appears a cleavage furrow. You can see the cleavage furrow on each side, indenting into the cell and dividing the cytoplasm of the cell also, so into two daughter cells, which is called as cytokinesis. So this furrow gets deepens and separates the two daughter cells. This procedure is called as cytokinesis. So totally these are the phases which are happening during mitosis. The significance of mitosis is, first thing, genetically stability. So it ensures the continuous succession of identical cells through generations. So as the parent cell having same number of chromosomes are given to the daughter cells. And growth and development also, that is it helps in the growth and development of the body. And the next thing is about the degeneration and regeneration and replacement of the and repair of the degenerated area. So it helps in the regeneration of new cells to replace the dead and damaged cells. So that is the significance of mitosis. Next we shall see about the reduction type of cell division, which is called as meiosis. The meiosis is a special type of cell division that takes place only in reproductive organs to produce gametes. So only during gametogenesis we find this meiosis. Mitosis is seen in somatic cells in other places also, like even in germ cells also we find mitosis. But meiosis only happens during gametogenesis. So meiosis consists of two phases of cell division that takes place one after the other. That is first meiotic division, also known as reductional division. In this, the number of chromosomes of daughter cells are reduced to half the number of mother cells. So it receives the half number, that is haploid number of chromosomes. Next, it is continuously with it with consecutive second meiotic division. In second meiotic division, it is similar to mitosis, but similar to one described above, like no duplication of DNA during, that is the major difference between the mitosis and second meiotic division. There won't be any duplication, no duplication of DNA during short interphase. And so that was about the second meiotic division. So let's talk about the first meiotic division. So first meiotic division we can divide into several phases. Prophase of the first meiotic division is very long and complicated, and it is therefore again divided into 5 stages, that is leptotene, then it is divided into zygotene, then pachytene, diplotene, diakinesis. So these are the 5 stages of prophase of first meiotic division. So during leptotene, in this stage, the chromosomes as in mitosis appears to be slender thread-like. We can see they appear to be slender thread-like, and then although each chromosome consists of 2 chromatids, they are joined at the centromeres, and the chromatids are not visible at this stage. So that is about leptotene. Second, about the zygotene. In this stage, the lengthwise pairing of the homologous chromosomes begins. We can see the chromosomes align homologously, lengthwise pairing, and one of the two homologous chromosomes is from the father, that is the paternal chromosome, and the other one is from the mother, that is maternal chromosome. So this event is called as synapsis, and each synapsing pair is called as bivalent. So that is about zygotene. Next, regarding the pachytene, this stage is very long and may extend even for years, and it is characterized by like the chromatids of each chromosome becomes visibly separate. We can see the chromatids becomes each separate. Here they are not separated. We are not able to distinguish the chromatids in leptotene and zygotene, but here we can see they are separated, and each bivalent chromosome thus appears to have 4 chromatids called as tetrahed. You will see a centromere and 4 chromatids on each, so it is called tetrahed, and each chromatid paired to a united by kinetochore. So you can see the central kinetochore. There are 2 central chromatids and 2 peripheral chromatids. 2 are towards the center and 2 are towards the periphery. So one from each chromosome. So the 2 central chromatids, one belonging to each chromosome of tetrahed, coil over the other. So the central one we can see they coil over the other, they cross. We can see crossing over. So due to the crossing over the central chromatids presents a cross link. You can see the appearance of cross link configuration called as chiasmata. It is called as chiasmata. So these crossovers are chiasmata. So next we shall discuss about the diplotene. Diplotene is characterized by the paired homologue of tetrahed starts separating, and during this process the central chromatids break at the point of crossing over. So at the crossing over they get break, and the crossover they may go to the opposite side. So there will be exchange of genetic material happens between the chromatids. So the central chromatids break at the point and crossing over and unite to the opposite chromatid, and this results in the exchange of genetic material between the chromatids. And lastly, the diakinesis, the chromosomes between more contracted and migrate towards nuclear membrane. We can see they are migrate towards the nuclear membrane, and at the end of the prophase the nuclear membrane disappears. The nuclear membrane completely disappears at the end of prophase. So that was about the prophase. Metaphase, the homologous pairs of the chromosomes becomes, see this is the metaphase one. So homologous pairs of chromosomes become arranged on the equatorial plate. You can see they are arranged on the equatorial plate and attached to the spindle fibers. And next is the anaphase. In this stage, you can see the homologous chromosomes migrate to the opposite poles of the spindles, and unlike mitosis, the chromosomes move randomly. The shorter chromosomes move earlier than the longer chromosomes. Next, regarding the telophase, this stage presents the features like nuclear membrane is formed around the paired group, that is polarized group of chromosomes, and the cell membrane constricts to two daughter cells. We can see the cell membrane constricts to form two daughter cells and are formed, called as cytokinesis, and each daughter cell is formed containing only half the number of chromosomes, called as haploid number, and with exchanged genetic material because of crossovers here. And next it continues with the second meiotic division, and second meiotic division is essentially similar to mitosis, and it is however differs from the mitosis in that the DNA does not duplicate. So by the end, by the second meiotic division, the two daughter cells of the first meiotic division from the form four daughter cells. So they will each having haploid number of chromosomes and haploid amount of DNA. So here haploid number of chromosomes but diploid DNA. Here the haploid number of chromosomes and haploid amount of DNA. So that is the final second meiotic division. So this completes the meiosis. So significance of meiosis, it is quite important in sexual reproduction. So as the chromosome number is reduced to half during meiosis, each germ cell has a haploid number of chromosomes, and when the two germ cells unite to form a zygote, the number of chromosomes will restore to diploid. And thus because of meiosis, the chromosome number is maintained for the species. That is the importance of meiosis. Then genetic variation. The second is the genetic variation, and because of random assortment of paternal and maternal chromosomes, especially during diplotin pachytene, we can see the random assortment of paternal and maternal chromosomes and exchange of genetic material during crossing over and in the meiosis. So the daughter cells, that is the gametes, have a new genetic configuration leading to a genetic variation. So this causes individual variations within the species. So we are not totally identical as our parents. We have some our own unique features. So that is because of crossing over of maternal and paternal genes, and this causes the individual variations within the species, which is very much essential for evolution. Next is the hybrid vigor, so which helps to maintain the vigor in the progeny through sexual reproduction. So these are the significant statements of meiosis. Next, let us see some major differences between mitosis and meiosis. Mitosis takes place in somatic cells, meiosis takes place in germ cells, and mitosis completes in one sequence. It completes in two sequences, meiosis 1 and meiosis 2, two successive cell division. Crossing over of chromatids does not take place in mitosis. but crossing overs of chromatids takes place during pachytene in prophase of one of meiosis 1. Then daughter cells are identical to each other as parent cell. Then daughter cells are not identical to each other to the parent cell, and equational division, it is a reductional division. So these are the major differences between mitosis and meiosis. So this completes the cell cycle and cell division, which is the first topic of today in embryology. We shall continue with the next classes, take classes of embryology. Thank you.